So we are back at the studio. We have another great interview for you. This is one I've been waiting for and planning for quite a while. So in the studio here, I'm Morton. I'm leading this EVIC TV today, and Mary O'Keefe, my colleague, will be around having interviews later today as well. But in the studio now with us, we have Associate Professor Massieh Moayedi.
Yep.
Yeah?
Yeah.
Sorry, it's always a bit difficult with their names. And so you are an Associate Professor at the University of Toronto, but you're also the Canada Research Chair of Pain Neuroimaging.
Yeah.
It's really specific though.
Yep.
But it does really well describe what you are all about.
Yes.
And I would say Mr. Pain Imaging, Pain Neuroimaging.
I mean, I'm one of a big group of us.
Yeah.
I also lead the SIG at ISP in pain neuroimaging.
Yeah. So SIG is a special interest group.
Yeah.
Yeah.
I think I have a responsibility to talk about this.
And we just had a brief chat now, and I said to you, I'm quite skeptical because pain obviously is a human experience.
Yeah.
And I assume you'd agree that neuroimaging is a wonderful technique to use as researchers.
Mm-hmm.
But it's also something that has been developing over quite a while, and I think we'll come back to that. But before we talk about neuroimaging, let's talk about you.
Okay.
What's your background and how did you become interested in pain research?
So I did my undergrad in biology. I developed a facial tumor when I was 19. And then I had a resective surgery, which gave me neuropathic trigeminal pain. And then I was curious about what caused that and how I could address it. And then I sought out graduate programs that investigated pain. I came across Karen Davis's work, and I was really interested. I applied, lucky to get in, and that set me on my trajectory to study pain. I did my PhD studying orofacial pain or temporomandibular disorders in Toronto for 5 years, and then I did a postdoc at UCL in London with Gian Domenico Ionetti, and now I'm back in Toronto as a faculty member.
Yeah. Good stop over with Giando.
Yeah.
And when we talk about imaging of the brain, as it were, and pain, and obviously you have the lived experience, so you would know about the experience, we'll come back to that. But when we look at the brain, mm-hmm. in sort of historical terms, what have been used to study the brain? Do you have any idea?
The earliest evidence comes from EEG, right? So that's over 150 years old.
And that's electroencephalograms,
that's correct.
Is that what it's called?
Electroencephalography, yeah. So you measure the intrinsic electrical activity of the brain, which is easy to do because the brain communicates in electrical signals.
Yeah. For people who don't know, so it's where you get that sort of shower cap on with lots of electrodes on it, and it registers where your brain's activated.
It's more of a summary measure through the skull, but it does look at activation in electrical signals in the brain. A newer version of that would be magnetoencephalography, which measures the magnetic waves created by electrical signals, but theoretically it's the same idea. And then more recently, in the 90s, we developed magnetic resonance imaging, which then got more advanced from structure to function, where we could look at bold imaging in the brain. So the blood flow or the oxygenation of the blood in the brain.
Yeah, let's just pause there because there's a lot of technicalities here, and I think it would be nice to have someone like you to explain it to someone like me.
Okay.
So I don't know the first thing about physics.
Okay.
But what I know about scans, and we can start with the MRI scanner. So the BOLD, blood oxygen level density scans.
Dependence. Blood oxygen level dependence.
Dependence.
Yeah.
So what you do there, literally, is you have people lying in the scanner, and then you measure the area of the brain, where there's blood during a specific stimulus, or is that how you do it?
So the fundamental assumption of BOLD is that when you use a brain area, it requires more metabolic function, and so there's an increase in blood to that brain region. And what we measure is a ratio of oxygenated to deoxygenated blood. So when there's an influx of blood, there's a rise of the
hemoglobin
blood.
is what the scanner really detects, is that?
Yeah, so what we're lucky about is that oxygenated and deoxygenated hemoglobin have different magnetic signals or susceptibilities. And the magnet can pick those up, and then we can measure this ratio.
So when you get this blob picture, I guess that is the technical term, isn't it? Blobs?
The blobs,
Lips and blobs?
Yeah. Clusters is more formal
so
now.
you get this colourful picture.
Yeah.
That shows you a gradient on how much blood there is versus deoxygenated blood. Is that what?
In theory, yes.
Yeah, in theory.
Yes.
And then that theory suggests that when there is more blood, the brain is more active.
Yes.
And then where do you go from there? I mean, how do you know what area of the brain does what? Are you working on a hypothesis, or are you just sort of experimenting and see what?
So MRI doesn't exist in isolation. What we do is we tie it back to the literature, right? There's centuries of literature where there was brain stimulation and lesion studies that have helped us understand the function of different brain regions.
So lesion studies would be like Brotman's area, where you have people coming in who cannot speak, and you find out there's something wrong in their brain, and then you do reverse engineering, as it were, saying this person couldn't talk after an insult to the brain or after a clot or whatever.
Yeah.
Therefore, this area of the brain is related to speech.
Yes.
So that's
precisely.
Yeah.
Another example is a famous neuroscientist or neurosurgeon from McGill, Penfield, used to take a large electrode and stimulate brain regions and see what would happen and ask people to report. So
if people don't know Penfield, they know the homunculus, right? So Penfield made the sort of famous homunculus of the brain studying. So he was doing neurosurgery, right, and finding out where not to cut in the brain. Was that it?
That was part of it, but it was also a mapping exercise as well. So they would literally put a large electrode on different parts of the brain and see what happens.
So you have the Penfield studies suggesting that, let's say, the lips are big and they have a representation in the opposite side of the brain.
Yeah.
So now you do a scan and you see there's more oxygenated blood, or you suppose there is, in this aspect of the brain where Penfield said the lips are.
Yeah.
Therefore, the lips are now part of the experience. Is that how we think about it?
That's part of it. The other part of it is that you very carefully design an experimental study where you repeat a stimulus over and over again versus hopefully a control stimulus, where you can then compare the activation. So one of the things our graduate students do very early on is a simple lab experiment where we have data of someone wiggling their toes in the scanner, wiggling their fingers, and then blinking. And what you get is that Penfield map in the brain of when they wiggled their toes, wiggled their hands and or fingers, and blinked. And you can very clearly see the delineation of these three regions in the brain.
Oh, okay.
Yeah.
So as long as they're sufficiently distant.
No, you can even catch right next to each other.
Okay.
So there are beautiful techniques now where, and this has actually been used in some chronic pain patients, to map out the digits. So as long as you do a proper study where you know what you're doing and it's well designed, you can literally map out the various digits in the hand and see how that changes in some chronic pain disorders like complex regional pain syndrome.
And I think we should come back to those patients in a minute. But, so we had BOLD. So first we said structural MRI, so you could see the brain, as it were. Then you had the BOLD we just talked about, and then there was the ASL. Was that the sort of next level?
Yeah, ASL.
Arterial spin labeling, I think it's called.
Yeah, so arterial spin labeling is a lot like, we skipped this one, positron emission tomography.
Yeah, okay.
So positron emission tomography is when you put a radionuclide or a radiation, a molecule that emits a radiation that a detector can pick up.
Are you putting that into the bloodstream?
You are.
Yeah.
And
so
there are
injecting into the bloodstream.
Precisely. And there are different types of radionuclides you can use. There are some that just measure blood flow.
Yeah.
So you tag it to water. Water is the most common element in blood, and then you measure blood flow. The other would be to do particular ligands or molecules in the brain to study systems. So one of the most famous is an opiate ligand. So you can put an opiate ligand in the brain and see where opiates connect during pain, for example.
So an opiate ligand would be...
Raclopride.
Yeah, okay.
They're just basically different... They're molecules that bind to the receptors in the brain.
To the mu-opioid receptors, or any of the receptors, opioid receptors.
Kappa, delta.
And by doing that, you get a visual mapping of where those receptors are in the brain.
Precisely. And you can also get a measure of when they bind more.
Okay.
So depending on the enzymatic activity.
So you still get a functionality of it when they bind.
Yes.
And the affinity of it, so how many there are?
No, the affinity depends on the molecule.
Okay.
But the density, absolutely, you can measure.
Yeah, okay.
So we do the same thing now with endocannabinoid studies, for example.
to find out where the endocannabinoids act on the brain, as
Right.
it were.
So you
But
can
you
measure
still,
out the receptors. You can.
I guess that wouldn't help you with Penfield's.
No. So Penfield's homunculus is functional.
Yeah, exactly.
Right. So it's
where do
more
you
of a-
where you correlate that? How do you map that towards or against what? So now you know that this area of the brain, let's say whatever the medial prefrontal cortex or whatever, you have these receptors here.
Yeah.
How do you understand that? Because in itself, as you said, in isolation, that is really not helpful.
No, again, you tie these to different paradigms, right? So I personally think that neuroimaging is largely useless if you don't have a good experimental paradigm, right? So tying a neuroimaging experiment with a theoretical framework that is then used to develop an experimental paradigm is how we develop clear, concise answers to answer questions. So going back to Penfield's homunculus, if you want to know how things are mapped out in the brain, you can't just stimulate the whole body at once. You have to very carefully design an experiment, understanding the constraints of the technology you're using. So when we're mapping out the fingers, you're correct, the fingers are very close to each other, you're not able to see them apart. And if I were to stimulate the thumb and the index, that'd be very hard to see. So what I do is I repeat the stimulus a very large number of times, but the other thing is I time them in a way that I can then map out that timing in the brain. And then I can dissociate these two activities. And that makes it much easier.
And then the information you get out is, that would be a number of zeros and ones essentially.
Yeah, or a gradient between 0 and whatever that number is.
And then how, so I'm just wondering, would it by now be something you would use algorithms to solve, or are you doing deep learning? Or
all of brain imaging, it relies on statistics, right? So everything, effectively when you're looking at a bolt signal, you're looking at about a 2% change in signal. So it's a very, very noisy signal with a very incremental increase.
Yeah.
But we have sophisticated algorithms that can really, really drill down to identifying those different changes in signals and where they occur in the brain.
So just coming back to the endocannabinoids and the functional paradigms that you would use. So you would have a paradigm of, would that
you
be
should come to my talk this afternoon. It's
Sorry?
all there. We're talking about that in my talk this afternoon, actually.
And so tell us, what's the paradigm you mirror it to or you compare it to?
So ours is, right now, because endocannabinoids have largely been understudied in pain, we're really starting at the basics. So we're giving people a heat pain stimulus and trying to see where that activity occurs, which we know what to anticipate. And then we correlate that with circulating endocannabinoids, and we're able to then identify brain regions where the concentration of endocannabinoid in the blood correlates with the activity in the brain.
So is that a classic pre-post design where you stimulate someone and it hurts so much and then you give them cannabinoids?
No, we're looking at the endogenous endocannabinoid system.
Okay, yeah,
But
okay.
you can also do the pre-post with cannabinoids, like it's been done with opiates and every other drug in the world. The advantage of studying endogenous systems is that you can then measure what's going on in the brain. The limitation, of course, is that we're using a peripheral measure, blood, and correlating to a central measure, brain activity. Ideally, what I could do is put a needle in the brain, measure your CSF, and measure that.
Depends on who ideal it is.
Right, and I would never do that, right?
Yeah.
But we do have radioligands for various brain markers of the endocannabinoid system, so the receptors and the enzymes.
Yeah.
And we can look at the distribution of those and the function of those in various paradigms.
And are we still talking PET?
Yes, that would
That's
be
the
PET.
same, okay.
And the issue there, of course, is threefold. One is the half-life of the molecule you're looking at. So your paradigm is very, very much reliant on the half-life of the molecule. The second issue, of course, is when you look at these measures, the resolution's quite low. You're not able to capture the high resolution that you would with like MRI. And the third issue, of course, is huge amounts of individual differences. So some people there are huge variabilities, like in all systems, and so it makes it very difficult to capture or a meaningful mean.
Yeah.
Yeah.
And are you always, so again, you use statistics, so you're always using means, so you're not looking at inter-individual changes?
So that's becoming more and more popular.
Yeah.
It's just, that costs a lot more, right? Getting a large enough sample to pick up individual differences requires a much, much larger sample. MRI is a technique that's quite expensive. And so these things add up.
Yeah. Oh, there's so much here in this. So let me just skip on to the ASL, the arterial spin labeling, which is essentially a similar type of thing, isn't it?
It's exactly like the PET imaging that measures blood flow.
Yeah.
So ASL tags your blood in the carotid artery as it enters the brain with a magnetic flip.
But when you say tag, what you do is you stimulate it with a
magnetic
radio
flip.
frequency. Sorry, what do you call it?
It's a radio frequency pulse. So what you do is, by sending in the pulse, you flip the protons in the blood. The protons in the blood literally means the atoms of water in the blood are given a specific magnetic spin. They're flipped a certain way. These protons want to go back to the way they were. When they do that, they release a certain frequency of energy that we measure. So what we do is we flip them, they go into the brain, they flip back, and then we measure that flipping back. And that lets us identify where that blood is going and where it's concentrating. And again, the assumption there is this idea of neurovascular coupling, which is this idea that as you get increased blood flow to a region, it's because there's increased demand for blood flow. Although I will throw in, and you can cut this out later if you want. I don't know if this is
it's live, so we can't cut anything out.
Okay. Is that there's a really cool study that was published in Drosophila, which are tiny little fruit flies.
Yeah, fruit flies.
That shows that actually the blood flow precedes the demand based on physiological memory of what's needed.
Okay,
so you can actually predict
predictive coding type thing.
That you can predict the behavior before it occurs, which is revolutionary, but very, very concerning for concepts of free will and so on and so forth.
But so we're going to get into theories in a second.
Yeah.
Are there new developments? So ASL, is there anything newer than that?
There's always newer technologies. One of the, not for functional imaging, but for structural imaging, I think Francesca Fardo here has some evidence on this, of really cool, super high resolution MRI that allows us to detect the molecular structure of the brain. So one of them is the concentration of iron in certain brain regions, or the concentration of myelin in certain brain regions, that then allow you to understand the physiological basis of structural differences and potential behavioral differences.
Yeah.
But the field is moving, in my opinion, towards higher resolution imaging. So 7 Tesla MRI is becoming more and more popular in humans, and I'd say one of the advantages we have with neuroimaging is that one of the big gaps in the literature is translation from rodents to humans and from humans to rodents. And there's been a real big push to really look at rodent imaging to try to reconcile what we see at the molecular level to what we see at the MRI level.
What about spinal imaging?
Spinal imaging is getting more and more popular. I will say something controversial here, and that it needs a lot more work to be much more convincing.
Yeah.
The statistics aren't quite there. I mean, if you look at the spinal cord, it's that big compared to the brain, which is this big, right? So when you're looking at the spinal cord, the resolution at 3 Tesla isn't quite there to really be able to drill down to what you're looking for.
And just for people, so Tesla is the measurement of how precise or how accurate,
how strong the magnet is.
The strong the magnet. So, and again, how precise you get, how detailed information you get out of it. Essentially.
Yeah, it has to do with the detail and also the resolution of the image.
Could it be dangerous at some point? So let's say Tesla 20 or something.
I mean, so right now 7 Tesla has been approved for humans. One of the things that happens is that as you enter a magnetic field, you can cause random firing in the brain. And that random firing can elicit nausea and a bit of disorientation. So there's also 9.4 Tesla and 11.4 Tesla imaging that's occurring. But those are more approved for rodents and animal imaging, not so much human imaging.
Okay.
Some people are working towards 9.4 Tesla for humans. I'm not convinced it adds that much more. 3 Tesla, which is the standard at the moment, is kind of a sweet spot for signal to noise ratio. So it's where we get the most signal for the brain. Some people argue that 7 Tesla actually has lower signal to noise ratio, but you can get higher resolution images. I leave that to the physicists. They do the work, I just apply it.
Nice.
Right.
So you have the paradigm, so you expose the person to something, or you have Penfield's idea of where things are, and you can sort of reproduce that, so you have some older evidence and you have some new evidence and you correlate that.
Yeah.
But also, I assume if you're doing experimental research, you need a theory.
Yes.
Could
Hopefully.
could we go through the theories and maybe start so the neuromatrix idea of that specific areas of the brain, maybe individual areas, but specific in the sense that they are there and they are activated at a point and they give rise to consciousness or pain. That's probably not where we are now, but is that fair to say that's sort of how it started?
So, for the longest time, people thought pain was peripheral, right? And what helped us understand that pain was central was imaging, right? Imaging really leapfrogged us forward and helped us understand that pain is a construct of the brain, and it is the integration of these various domains of the brain that bring this together. This was first proposed by Massieh and Wall in the 60s, but Massieh and Casey, in the 70s. But that really took off with imaging that helped us see that. Let's back up a bit. The theories of pain have existed for millennia, but they really became formalized in the 20s, where we had this idea, even in the late 19th century, where we had this idea of two competing theories, labeled lines, specific pathways that are pain specific, and then pattern theory.
So a pain pathway, I suppose.
A pain pathway from the periphery all the way to the brain.
Yeah.
And then there's this idea of a pattern theory, where you have different patterns of activations that signal pain at the level of the brain. And then there's the third competing idea, which is this idea of a regular pathway that then, if you give too much input, it then becomes painful. In reality, all three ideas have their valid aspects, right? We do have nociceptors that are pain-specific receptors that connect to nociceptive fibers, that connect to second-order fibers in the spinal cord that go to the brain. So specificity absolutely exists peripherally. But that falls apart when you get to wide dynamic range neurons in the spinal cord, where you have input from innocuous and noxious fibers, and somehow the second-order neuron needs to know the difference.
Yeah.
And then when you get to the brain, it all falls apart, because there are very few nociceptive-specific fibers in the brain. And I guess
this would be posed, maybe, but it's subcortical, or maybe the thalamus is sort of
even
the
before
endpoint
that,
of specificity, wouldn't you say, or is there?
Even before that, you have input to various spinal cord regions. You have parallel. You have about 5 parallel pathways that carry nociceptive information to the brain, but how that's coded separately in these different pathways is unknown. You have thermal information and nociceptive information carried up the spinal cord and to the thalamus, but also various brainstem regions as well. So how do these regions tell apart these different inputs remains an open question. And then from there, from the thalamus, you go to different brain regions. So S1, primary somatosensory cortex, secondary somatosensory cortex, the insula, the cingulate, the prefrontal, the amygdala. What you find with brain imaging is that most of the brain is actually quite active in pain. And I think one of the advances of brain imaging and really clever designs has been the dissociation of all these different regions, and not seeing it as a unitary function, but seeing it encoding different aspects of pain.
Is that, again, that's the skeptic in me saying, is that any different if you were studying stress or disrupted sleep or extreme joy? Are those regions not involved in that as well? Or
it depends. This primary somatosensory cortex doesn't respond to extreme joy, stress, or joy, right? So you have the primary somatosensory cortex. The other thing is, one of the key networks in the brain involved in nociception and pain is the salience network.
Sorry,
the salience network.
Yeah.
But as the name implies, the salience network responds to anything that is salient. And salient just means...
I guess maybe the default mode network first, and then the salience network.
I would argue that.
Okay.
Sure, we can talk about the default mode network. Let's back up a bit again, and talk about what we think about the brain.
Yeah, so the neuromatrix model was there. There was sort of evidence suggesting that couldn't explain exactly how the brain might work.
There's a neuromatrix model from Massieh, which is very different than the pain matrix model, which no one believes in anymore, which is a shorthand now for nociceptive regions and pain modulatory regions, which has now become the dynamic pain connector. But these are all just shorthands for a very complex set of regions that work together. Moving back to the neuromatrix, what Massieh was proposing was that there are different aspects to pain. Pain is a complex behavior that is not just sensory or affective, but it really involves a complex response. So there is the sensory aspect, how much does it hurt? The affective aspect, I don't like this, I need to get away from it. The motivational aspect, I need to do something about this. The decision-making process, why is this happening? Should I do something about it? What should I do? And then the motor response. And somehow this entire behavioral repertoire needs to be mounted and occur. And so what Massieh was saying is that this isn't like a simple process of like sending a light to a receptor in the eye that then lights up your visual cortex. It's more a complex experience, like saying an apple, right? An apple has color, it has shape, it brings back memories, you have to contextualize it. You have to then remember the taste and the fact that it's a food. And all of these different things really help you bind this experience to then say, this is an apple. And the same is true of pain. It's not a simple sensory experience like activating a single nociceptor. It's really, what is this stimulus? Is it harmful? What does it mean? Why is it happening? How do I respond to it? Have I experienced something like this before? And all of those things have to come together to then elicit a response.
And this is, of course, the pain experience, right?
Yes.
And then coming back to the brain imaging. So post
So this was proposed. This was proposed by Massieh. There was no way to show that before brain imaging. So brain imaging helped us understand each component of that pain experience, understand their distinct encoding in the brain for each of these experiences.
And I guess in time, so this is back maybe 15 years ago.
Yeah.
Just looking at the brain, everyone was amazed, and then time went and studies came out like Andy Moreau and Ionetti's study on the pain matrix, maybe more salience activity system, and also the frozen salmon study, obviously.
Yeah. Well, I mean, so here's the thing.
Just building in a bit of criticism, I assume.
Yeah, I mean, I can criticize any technique with bad statistics, right? So the whole point is, when you do a brain image scan, you're scanning 100,000 voxels in the brain, right? You need to correct for each of those statistical tests. And the dead salmon study that you're discussing, for those who don't know, is that they put a dead salmon in the MRI scanner, and you had activation in the brain. But MRI, like any other signal...
Well, you didn't, but you had MRI results suggesting there was activation.
Right.
Yeah.
Right. There was
we assumed there wasn't.
There was a voxel that lit up in a series of voxels. But like any signal detection method, there is noise in the system.
Yeah.
There's always noise in the system. And so what we do with statistics is that we assume that we're comfortable with a certain threshold of noise, and that we believe that the signal that we're measuring is above that threshold of noise.
And was that then the sort of, at least that's how I understood it, that was sort of maybe the stepping stone into this idea of a default mode network where there was...
No, not at all.
Okay.
No. Sorry, there we go. So what Ionnetti and Moreau showed, which is actually why I joined Jando's lab, is that the activation you see in the brain, that is the typical brain activity that you see, is not pain specific, but reacts to any modality that is attention grabbing. Now, that's not entirely true, because S1 does not respond, the primary somatosensory cortex does not respond to any modality, it responds to sensory input. But that was more a criticism of using a shorthand for brain activity related to nociceptive input and pain. And it's also a criticism of the lack of experimental rigor in the study designs that led to that shorthand, right? And I think that that's where most fields are self-correcting. I mean, you can look at the rodent literature. I was just in a talk about an incisional model of pain in rodents, And people were saying, well, you only measured this one modality. What about these other modalities? What was your control? And I think what it is that we moved on from the early days where everything was low-hanging fruit. We had no idea what pain looked like in the brain. We didn't know what was going on, to one of scientific rigor where we're like, hey, we now have the low-hanging fruit. We understand what's going on. Now we need you to really think about what you're doing more carefully and design studies that capture what we now understand about pain. So we used to say the pain experience was universal, and now we understand and accept that the pain experience is vastly different for every single person, and we need to look at those individual differences, right? We used to accept studies in rodents with 2 or 3 mice. We don't do that anymore. We used to accept clinical studies, clinical trials with 12 patients. We don't do that anymore. And the same is true of imaging. Now, imaging is constantly criticized as being a very expensive modality, but I'm sure you haven't looked at optogenetics and the cost of breeding a mouse that can respond to that as well, right? So at the end of the day, what we're doing is we're really trying to refine our techniques to improve the reliability and the robustness of what we find.
So coming back to the default mode network.
So the default mode network was first discovered by, I believe, Biswal in the 90s. And what it was he was looking at a simple motor tapping task, and then he looked at the regions when people weren't doing something.
Yeah. So in a relaxed
off
state,
period,
as it were.
the relaxed state or the resting state. And what he found was that there was actually a series of brain networks or regions that were constantly active when the rest of the brain, when you weren't doing a certain task. This later became more formalized in the early 2000s.
So the person was relaxing, but the brain was still active.
Right.
Yeah.
And this was quite revolutionary because we always thought the brain responded to stimuli, was like a passive organ that responded to stimuli. And this really forced us to reevaluate that and say, wait. The brain's actually involved in a lot more. And it's not just doing this background tone activity, it's actually constantly evaluating the environmental milieu. So what they called this was the default mode network of the brain, which we then at the time thought was involved in internal processes of the brain and monitoring of the body. Moving forward now.
Do we need to change the microphone? Yeah.
Okay. Sorry. Yeah. Okay. What we're finding now is that in fact, what we thought was internal monitoring of the milieu is probably more involved with 2 different memory networks. Because the default mode network, if you break it into 2 cores, really falls into the episodic and the autobiographical memory networks.
So the theory itself was that there's a brain activity when you are resting.
Yes.
And then whenever you are exposed to stimulus,
up and down.
that extra is what we would then suggest is related to the stimulus. Is that right?
Yes.
So you lie down and your brain is active, and then you have a stimulus that is experienced as painful, and you would say the areas of the brain that are involved in that are then you're sort of subtracting you're subtracting the resting state, as it were.
Yes, in theory. Now the reality that we know now is that these networks are humming along up and down, and that the perception of a stimulus relies entirely on, sorry, this keeps rolling. The perception of a stimulus relies entirely on the state of the brain when it's received. So, if you're in a state where
is this individual or is it per state?
So in general, if a network is low and you receive a stimulus related to that network, you're going to perceive it less than if that network is high and primed to receive that input.
And just to clarify, so when you use, so you're reusing the same word, but essentially you're meaning something else. So a network back in the default network was aspects of the brain doing something, and that's the same thing, but it's not necessarily the same regions of the brain. Is that correct?
So, let me be more clear. There are various networks in the brain. They're called intrinsic connectivity networks.
Could you give an example of a network?
The default mode network, the salience network, the visual network, the somatomotor network. These are all networks that involve regions involved in different functions. And these regions, what we mean by network is that their activity is correlated and they're temporally correlated, so they go up and down together. So when your brain is at rest, these networks are humming along. They go up and down in rhythmic motion. And what we're finding is that as they hum along, different networks peak and go through peaks and troughs. if you give someone a stimulus, let's say your visual network, you give someone a visual stimulus that's barely perceptible. When the network is in a trough, you're less likely to perceive it than when the network is in a peak.
Okay.
Right? So this is
So
how
if you already have occipital lobe activity, you're more likely to perceive something that is of low light. Is
yes.
that how, yeah?
You're more primed to receive something
Does that then
activity.
translate into experimental pain, for instance?
Absolutely.
If you are expecting a stimulus to be painful, then even...
It's not even expecting. It's the time at which you stimulate a person and the state at which their brain is when you stimulate them.
So the brain state is not necessarily the same as the conscious state? Is that attention state?
No.
So this brings us into one of your theories.
Yeah.
So you with Karen, you wrote an article a few years back, suggesting, and maybe 10 years back, to be honest.
Oh wow.
Suggesting that there was,
so
I'm paraphrasing, there was a switch, a gate, as it were, in the brain. So there was like there was an activity that could be painful, experienced as painful. It was in the brain, as it were, but it wasn't switched open, or there was an area of the brain that was shutting it down from being consciously experienced.
So that paper was more for to bolster discussion. We've kind of gotten in a stagnant time in the field where we weren't thinking really much about what is pain and where does it come from, right? Like there is this concept of nociception, which is the carrying of nociceptive information to the brain. But when does that nociceptive information get integrated to a pain experience is unknown. It's still unknown. I can't go anywhere in your brain and stick an electrode and make you feel pain. Now, Luis Garcia-Larrea, future president of ISP, or of EFIC, would strongly disagree with me, and so would Roland Perron, because they've stuck electrodes in the posterior insula and elicited pain. But what no one discusses is that some parts of S1 do the same thing. Right? If you go back to the earliest Penfield papers, 11 of 52 regions that he stimulated elicited pain. The reality is, there isn't enough experimental evidence to know when nociception becomes pain and how that occurs. One of the dominant theories is that there is a connection between the posterior insula and the cingulate, and it is a directional stimulus from the posterior insula to the cingulate that then elicits a sensation of pain. But there isn't enough empirical evidence to support that model.
And just to clarify, when you say the posterior insula, what we really mean is an area of the brain that has a specific characteristics. So it's, in this case, it's located a specific place, but it's also there's a specific affinity of receptors or something else that defines it. It's not purely anatomical, is it?
No, the dorsal posterior insula is purely anatomical. It is, the insula is a part, so if you look at the brain, it's got two halves. This half has the temporal lobe, and then you have your frontal lobe. If you pull these two apart, you find this island of cortex called the insula. Insula literally means island. So it is an island of cortex. It has variable shape across people, but in general, you have the anterior portion, the mid portion, and the posterior portion. Now, the anterior portion is involved in a lot of important regulatory functions, in eliciting a sympathetic and parasympathetic response, and detecting salient stimuli and mounting responses to salient stimuli. The mid portion, we're not quite sure what it does. Some people say it's involved in taste. We're not sure. The posterior portion is largely involved in interoception, or monitoring the body. But some people, like the late Bud Craig, argue that it actually was pain cortex. It is the only part of the body, or the brain, that receives direct nociceptive input via the thalamic pathways. It's still controversial. I think you'll find very many different opinions at this meeting. But the posterior insula, in monkeys, for example, has very few connections to the mid-cingulate cortex. There are no direct connections, actually, more than one or two fibers ever detected. So we just published a paper in Human Brain Mapping, in cerebral cortex, that was the first to show the connectivity of the posterior insula to the mid-cingulate cortex in humans. and really show that there's robust connectivity there in humans that's absent in rodents and in non-human primates. So really, this is really helping us understand what might be pain specific, but we're really nascent in that field.
And I guess that's what research is all about. It's exploring these opportunities and changes. But just coming back, so that when we say the posterior insula, what you are referring to is a region of neurons. How many neurons?
Tons.
So
many tons.
So are you then averaging a response in that area, or are you looking for anything within that sort of?
So the dorsal posterior insula is roughly the size of my fingertip, which is a huge part of the brain, and has, I don't know the density of neurons, but a lot of them, and it responds specifically to temperature and to noxious stimuli.
So is it like, so I don't know, let's imagine how many people in Toronto, 10 million people, or I don't know. So let's say there's 10 million people in Toronto.
Yeah.
Are they then all responding to the same thing, or is it as diverse as Toronto?
Absolutely not. It's as diverse as Toronto, right? But the idea is, until we can do layer fMRI of the insula, which is maybe a decade away, we're not really going to understand that, right? So don't forget that the brain comprises of at least 3 to 6 layers of cortex. Each layer has different functions. Some layers specifically receive input from the thalamus, other layers receive input from adjacent cortex, other layers receive long distance input, and other layers are involved in integration and feedback loops. And then some of the layers send back to the thalamus and other layers send to other brain regions. So every single part of the brain is incredibly complex. So when you look at S1, for example, S1 is set up in what we call columns. So in your skin, in your hand, you have 4 different types of receptors. You have Pacinian, Ruffini, Meissner, and I always forget this other one. Merkel. No.
Merkel, yeah.
Merkel.
So these are all low threshold sensory
low threshold
fibers.
mechanoreceptors. Yeah. They respond to different types of stimuli in the skin, and each one in every part of the body is represented in your somatosensory cortex. And each one from the periphery has a clear column of activity, and they're adjacent to each other. Now that's absolutely true for area 3A, 3B of the primary somatosensory cortex, but 3A receives input, excuse me, 3A receives input specifically from the deep tissues and proprioceptive input, where 3B receives input from the rest
I guess
of
it
the
was
body.
easier when there was a line labeling theory explaining everything, wasn't it?
Absolutely, right? But that gets completely distorted in pain.
Yeah. So I always keep coming back to the default mode network, but you were introducing also.
Well, we have at least 9 intrinsic brain networks.
Okay, but I guess the saliency network is probably the one we should mention here. And then I have a different question for you, sort of to finish off.
So there's a default mode network, there's a salience network, and there's a somatomotor network. These are 3 key networks that we know are involved in nociception and pain.
Which is why, so when you when you have humans experiencing pain, you would see these 3, so activation of the brain, that's associated with or explained by these networks.
Yeah. But these networks are involved in a lot of different functions. The question is, what is her role in pain? And again, because you're generalizing over tons of neurons and tons of brain regions, what you're going to say is that the salience network is involved in the salience detection of pain. Absolutely, sure. But, excuse me, what happens is they have differential functions and differential connectivity in different contexts, right? And that's why we need good experimental designs to be able to parse that out. If I give you a heat stimulus and I see an activation in the brain, I can say, yes, that's the brain activity to heat. Great. But that could be the brain activity to any number of stimuli. But if I have a proper, excuse me.
Oh, bless you.
I don't talk this much normally. Excuse me. But if I have a good control stimulus, then what I can do is start understanding where those where those activations differ between modalities. And it really comes down to, excuse me, understanding how those things vary across modalities and across stimuli. And one of the more important things, one of the bigger advances, I would say, is that we've really moved away from activation studies, where these are the regions that light up, to these are where the different regions of the brain talk to each other. I think that's where imaging is going, and understanding the network discussions between brain regions, if I were to answer Prof, that's where we can really hone in on what's going on in the brain.
Yeah. And I guess this sort of summarizes really well where we are at the moment, right? When we use, again, you mentioned heat or even sensory stimulus or visual stimuli, these are far away from the lived experience of chronic pain.
Yes.
So let's say this was imaging looking at experimental pain. Where does imaging, and this might not, this could be just a quick response because I don't know, but where does pain fit into imaging instead of imaging fitting into pain? So what if people have pain? What can imaging do, or where are we with that momentarily?
So it's a really interesting question because it's something we struggle with a lot too, right? Imaging, I think, is hugely important in pain.
And of course, you're not biased when you say that.
No, not at all.
No.
I mean, if all the grant funders are listening, make sure to fund this. Imaging is actually, in my opinion, really important in pain for three reasons. It's one of the few ways we can look at pain in humans, where pain occurs in humans, right? So it's the only way to capture the full pain experience and find its behavioral correlates. Two, sorry. Excuse me. Two,
that's the unfortunate thing about life. We can't just stop.
No, it's okay. It's just...
but you can cough all you like.
It is what it is. Imaging allows us to do that. Two, imaging has made us understand that the brain functions in networks, right? So it's really advanced we've really moved away from a regional activity of the brain to understanding that every function of the brain requires many regions to come together to integrate an experience, right? So you can literally flash a beam of light of a particular angle, of a particular thickness, and activate a single receptor. Much like I can put an electrode in your hand and activate a single nociceptor. But that in no way represents what a real painful stimulus looks like. But what we realize is that at some point that needs to be integrated in a more complex experience, and that's what this network activity does, right? So our understanding of pain has really evolved from it's a peripheral disease to it's a disease of the CNS to it is related to the network activity of multiple brain regions coming together, and that pain is an emergent property of this network activity. So in other words, you need a lot of brain regions to work together for you to experience a pain experience. Now, someone like Marshall DeVore would rip this idea apart because he says you don't need the brain for pain. In fact, there are cases of people who have encephalopathy who are missing much of the cortex who still feel pain. And I have a few arguments there. One, how do you know it's pain, right? You can have reflexive behavior without pain. Two, those people have maintained an ability to protect themselves. And there's a lot of plasticity that can occur in that window. And three, just because the brain is smaller doesn't mean it doesn't have those functions, right? So
and again, coming back to the injury studies or people who have been exposed to this, they are unlikely to have a nervous system that is comparable to someone who's not had it. Yeah. That flexibility is obviously going to be there.
And that's what we do, right? In our chronic pain studies, we investigate people who don't have chronic pain versus those who do have chronic pain. Yeah. But even those studies are limited now because the assumption there is that chronic pain is the same versus controlled are the same. And now we understand that there are huge individual differences.
What I wanted to come to is that essentially what we are trying to study is the brain as it is normally.
Yeah.
And then I guess most people would agree nowadays that there are changes happening in the brain with people who have chronic pain. We don't know whether or not they're causal, how much it takes, whatever, or even if it's a brain-driven disease.
Yeah.
But I guess I take from your last sort of argument here that we don't know where the chronic pain fits into the brain as it is.
No, so I disagree with that, right? I think what it is that what we're learning is that there are phenotypes of healthy individuals who are at risk for chronic pain. And I think what we're learning is that those individual differences are something we need to look at. So you can't group people into broad categories of like neuropathic pain versus healthy. Of course you can, and you can look at very large effect sizes that occur in these groups. But to start treating pain, you need to start looking at the individual differences of people. And so to do that, what you need are much larger cohorts that are much better phenotyped. And then when you do that, you can start parsing pain-specific mechanisms that occur and that can capture that variation across patients. So here's an example. My lab with Jakob Ochofi in Toronto, we look at, one of the diseases we look at the most is temporomandibular disorders, which is a type of muscular jaw pain that occurs in about 5% of the population, of which 12% of those people develop chronic pain.
And so it's a classic non-specific pain, in a sense it's specific where it is, but
It's
we don't understand the driver of it.
We don't understand the driver, we don't know where it comes from. We know that the people who suffer from it find it very horrible and painful.
Yeah.
We've recently shown using MRI, but not of the brain, that the muscles are different. Again, what came first, the pain or the different muscle? We don't know. What we are showing is that there are individuals who don't have pain, but who have behaviors that have been found to be strong predictors of the development of TMD, or temporomandibular disorders. And these people look more like the patients than they do like the people who are not at risk. And so what we need to start doing is being much more cognizant of these differences of low risk, high risk, and then the patient groups themselves. And that will
how
really
are you going to
help
get
us.
that through ethics?
We have it already.
Yeah?
Yeah, because we are able to predict who's going to develop pain. We've developed beautiful predictive algorithms based on behaviors. So, for example, in TMD...
behaviors, it would be mouth opening, or would it be brain behaviors?
So in TMD, for example, it's people who clench and grind their teeth during the day. If your means of dealing with stress is to clench your teeth while you're awake, that's a high-risk behavior for developing TMD. Now, if you're someone who clenches their teeth a lot during the day and you're highly anxious, then you're closer to a patient with TMD than someone without TMD. So one of the more elegant studies we could do is compare a patient who's high risk versus a patient with TMD, and then find what the difference is, because this person has pain and this one doesn't. And that can tell us what chronic pain looks like in a patient versus someone who's very similar to them but doesn't have the chronic pain phenotype yet.
So if I was to ask you as the final question here, is chronic pain a state of the brain? Or even more, you know,
I think it depends what kind of chronic
even
pain you're
sharper.
talking about.
Could a brain have pain, essentially? Could a brain change into a phenotype that is so close to what we understand about living experience of chronic pain?
I mean, that's the whole point of nociplastic pain, isn't it?
Is it?
Yeah. The whole point of nociplastic pain is that you have pain persisting beyond the injury, and the reason you have pain persisting beyond any injury is that you've driven plasticity in the central nervous system that then maintains the pain. So I think it depends what kind of chronic pain you're talking about, right? And I think this is one of the big mistakes we make when we talk about imaging and pain, is we lump all pain together. But if you're talking about musculoskeletal pain or neuropathic pain, very different phenotypes. And then even within musculoskeletal pain, you have the widespread pain, which is really non-specific and poorly understood, as opposed to like a shoulder injury, which is just a persistent pain in the shoulder. Now, do I think the shoulder injury is uniquely in the brain? No. There is obviously a peripheral nociceptive input that's driving the pain and driving nociplasticity that maintains pain. You fix the shoulder, you're likely going to get rid of the pain. Not always, but you're likely going to get rid of it. Other chronic pain diseases, like widespread pain, I think are resulting from brain abnormalities. There might be a triggering event that elicits the pain, but that doesn't mean that the pain is there anymore or the signals are there anymore. It's really more maintained by the CNS. We have a really wonderful paper from the UK Biobank that actually shows that a series of genetic abnormalities and early life adverse events set up the brain to be more likely to have pain and predicts the number of sites of pain you'll have. And then it was done more formally with a broader set of genes and pain stuff from Etienne Vachon-Presso's lab, which I think he's presenting this afternoon as well. But the idea is the brain does change, and then at some point can become the source of pain in some chronic pain states that are more complex than something like a bad knee.
You almost got out of it.
No, I think it depends on what you mean by the chronic pain state.
And that, of course it is. I mean, we don't know essentially, and it's likely to be something that we thought we knew, and a lot of things we didn't understand when it happened, and then we get all the wiser. I guess that's why all the funding should go to MRI scans.
Of course.
Yeah?
Yeah.
Well, good luck with that.
Thanks.
Yeah. Thank you so much for sharing all of your knowledge.
No,
it's been amazing.
I hope you learned something.
I did.
I learned a lot, actually.
Yeah, no, it's brilliant. So thank you so much for showing up. I hope people at home got a lot out of it. You can always re-listen. I think some of what you said was quite intense. And definitely, I'm going to go back and have another listen to it. That's a good thing about internet. You can just go back and find it again.
And if you get stuck on anything, feel free to send me a tweet and I can try to answer it, or an email if you don't want it public.
Perfect. And there are actually quite a good number of papers, brilliant reviews, going through the history, and as you said, there's a lot of things. So you've been quite good at communicating this stuff.
That's very generous, but thank you.
Thank you for spending your time here with us.
No, my pleasure, absolutely.
Thank you.